Welcome to the deep dive. Today we're doing something a bit different, tackling two really specific neurobiology topics from your research stack.
That's right. They seem pretty far apart at first glance.
Yeah, one's about how a fly brain gets wired up with incredible precision during development.
And the other is about how, in some people, just looking at a certain pattern, a static image, can actually trigger a seizure.
But the link, as you pointed out, is this idea of timing, precise temporal control.
Exactly. Whether it's building a circuit correctly or, a breakdown in how an existing circuit functions moment to moment, it's all about keeping time.
Okay, so our mission today is to unpack the mechanisms behind that timing in both cases. Let's start small, really small. The fruit fly, Drosophila.
Good place to start. We're looking at how a single type of stem cell, the neuroblast, manages to produce hundreds of different kinds of neurons.
Just one cell line doing all that work.
Well, one lineage type. Specifically, we're talking about type 2 neuroblasts, or NBs, in the larva. They're like neuronal factories.
And the big challenge is making sure neuron hashtag 5 is different from neuron hashtag 15, and so on.
Precisely. It's called temporal patterning. How does the neuroblast know when to make what? It needs an internal clock.
And the research you pulled highlights one specific gene as like the starter pistol for this clock, SVP, 7-up.
Yeah, SVP seems to be the trigger. But it's not just flipping one switch. It kicks off this really elegant interplay.
Okay.
It sets up a dynamic based on two other genes, IMP and CYP. You can think of them as setting the rhythm.
IMP and CYP. Should we remember the full names? Improper localization and syncrip? Or just think functionally?
Functionally is probably easier. Think of IMP as the early marker and CYP as the late marker.
Okay, early and late.
And the cool part is, the system ensures their levels move in opposite directions within the neuroblast as it divides,
So IMP goes down.
while CYP goes up. Simultaneously, they form these opposing gradients.
Like mixing two colors, but over time. The internal state of the cell is constantly shifting.
Exactly, it's a chemical gradient clock. And that changing internal state defines what kind of neuron can be born at that specific time.
Ah, so the cell passes through different competence windows, you called them.
That's the term, yeah. About 40 distinct windows, roughly corresponding to the 40 times the type 2 NB divides.
40 windows, 40 divisions. So each window potentially yields a different type of neuron.
That's the idea. It's how you get this massive diversity from a single starting lineage. Biological choreography, really.
And the numbers are pretty big, right? Around 400 cells total from one of these type 2 NBs.
Yep, in that ballpark.
Right.
And they've identified, I think the sources said, over 80 genes now, with specific temporal expression patterns tied to this process. It's complex.
It really shows how much instruction can be packed into the cell itself, but are all the questions answered here?
Oh, definitely not. This leads to some big open questions, perfect for you listening if you're thinking about future research directions.
Like, okay, if there are 40 windows needing 40 unique temporal states, but the core gradient relies heavily on just IMP and CYP plus a few others.
Right. The sources mentioned maybe 5 core temporal factors driving that main gradient. So how do you get 40 truly distinct outcomes? What fine-tunes each window?
Feels like there must be other layers to this clock we haven't found yet.
That's a major puzzle. And another one is, are all 400 or so cells genuinely unique? Or do some of these slightly different temporal windows end up producing neurons that are functionally pretty similar?
That makes sense. And the other big question, is this all happening inside the cell line? Or are there signals coming from outside? Extrinsic cues.
That's the hunt. Finding those would be huge, especially for connecting this fly work to, say, how our own brains develop.
The sources mention only one exception, right? Something called hedgehog, AJ?
Yeah, hedgehog is a well-known signaling molecule. But here, it acts in a very local way. It's described as paracrine.
Paracrine. Okay, define that for us. Not everyone's a developmental biologist.
It just means the signal doesn't travel far. It's released by a nearby cell, maybe even a sister cell within the same lineage group, and influences its neighbor.
So it's still an external signal, but very short range, almost like internal crosstalk within the broader lineage.
Kind of, yeah. It's not like a hormone traveling through the whole body. Finding more examples of any external input is a big goal.
Okay, so that's the fly brain intricate timing built in during development using these gene gradients. Amazing stuff.
Really is.
Now let's shift gears pretty dramatically from building the brain correctly to, well, what happens when timing goes catastrophically wrong in the adult brain?
And specifically, triggered by something seemingly simple, looking at a picture.
Right. This whole phenomenon of photosensitive epilepsy. You mentioned sources showing this isn't new. People noticed this ages ago.
Yeah, it's kind of wild. Back in Roman times, 2nd century, Apuleius wrote about watching a potter's wheel.
The spinning wheel.
Well, interestingly, the original Latin apparently emphasizes the pattern of the spokes, not necessarily the flicker or motion. It could make people dizzy or even trigger seizures.
So the trigger was the spatial pattern itself, a static arrangement of lines or shapes.
That seems to be the key point from the historical context, and it matches modern understanding too. It's not always about flicker.
Okay, so what kinds of patterns are we talking about? What features do these trigger images usually have?
The sources list a few common ones. They tend to be large, definitely high contrast.
Black and white stripes, that kind of thing.
Exactly. Or things like backlit window blinds, some types of mesh or grills, striped garage doors, often with intermediate spatial frequencies not too fine, not too coarse. And symmetry seems to play a role too.
So a simple visual input causes a temporal meltdown in the brain. How? What's the proposed mechanism?
The work by Hermes and others points to brain rhythms. Specifically, these problematic images seem to really strongly amplify gamma oscillations in the visual parts of the brain.
Gamma oscillations. Okay, need a definition. We hear about brain waves, like alpha, beta. What's gamma?
Gamma is the faster end, typically defined as rhythmic activity between 30 and 80 hertz, so 30 to 80 cycles per second.
And it's not just random noise, it has a function.
Oh yeah, it's thought to be really important for healthy brain function. Things like attention, sensory processing, binding different features of an object together.
How does the brain actually make these gamma rhythms?
It comes from a tight loop, a feedback interaction between the main workhorse neurons, the excitatory ones using glutamate, and the inhibitory neurons using GABA,
Okay, excitatory fires.
which then activates the inhibitory one.
Which then pump the brakes using GABA, right? They inhibit the excitatory one.
Exactly, they quench the activity.
Mm.
But then, as the inhibition wears off after maybe 20, 40 milliseconds, the excitatory cells start firing again, and the cycle repeats. Fire, inhibit, recover, fire.
And that repeating cycle creates the 30-80 hertz rhythm. Got it.
Precisely. Now here's the connection. What kind of visual input generates the strongest gamma oscillations?
Let me guess. High contrast, black and white, sharp focus, maybe oriented lines.
You nailed it. The very features that are known to trigger visual discomfort or seizures in susceptible people.
So the images that cause problems are basically pushing the gamma-generating circuits really, really hard.
It seems that way.
Yeah.
The thinking is that inhibitory connections are crucial for sharpening our spatial vision, helping us see edges and details clearly.
Makes sense.
So driving those inhibitory neurons hard with the right kind of pattern is super effective at inducing strong gamma. It's like hitting the resonant frequency of that excitatory-inhibitory loop.
But most people look at stripes and are fine. Why does this perfectly normal, even essential gamma rhythm cause a seizure in some individuals? Where does it go wrong?
This is where the idea of impaired feedback control comes in. Generating strong gamma is like a stress test for the brain's ability to regulate itself.
Okay.
In people with photosensitive epilepsy, that feedback control might be weaker or somehow compromised. The inhibitory system, the GABA system, might fail under that intense repetitive gamma driving.
Fail how? Does it just stop working?
It's even stranger than that. This is the really interesting part. Under conditions of massive, sustained GABA release.
Which is what's happening when gamma is driven hard.
Right. That intense GABA release can actually mess with the local chemical environment around the neurons. Specifically, it can lead to a buildup of potassium outside the cells and chloride inside the cells.
Okay, chemical imbalances.
And if the cell's machinery for pumping chloride out is overwhelmed or faulty, which is one hypothesis in epilepsy,
Yeah.
then the concentration gradient for chloride can actually reverse. So when GABA opens the chloride channel, chloride flows out instead of in.
Wait, if chloride flows out, that makes the inside of the cell less negative, which is
It's-
excitatory, not inhibitory.
The GABAergic synapse, the brake,
Yeah.
suddenly becomes an accelerator.
Wow. So the system designed to prevent runaway firing actually starts promoting it.
That's the proposed switch mechanism. It dramatically increases the overall excitation, leading to the uncontrolled firing we call a seizure.
And this isn't just theory, there's evidence.
Yeah, it's consistent with some optogenetic studies where scientists can artificially activate inhibitory neurons, and sometimes, paradoxically, that activation can actually trigger a seizure, likely via this kind of ionic shift.
That's a fascinatingly scary breakdown of a fundamental system. Okay, let's try and tie these two threads together.
Quite a contrast, isn't it?
Yeah.
Development versus pathology.
Yeah. On one side, we have the fly brain, meticulously using these IMSIP gene gradients like a clock to create neuronal diversity over, 40 specific time windows. Incredible developmental control.
Building complexity with temporal precision.
And then on the other side, we have the adult human visual system, where a simple spatial pattern like stripes can hijack a normal brain rhythm
The gamma oscillation.
and push the inhibitory feedback system so hard that it flips, turning inhibition into excitation and causing this destructive temporal resonance, a seizure.
A loss of control triggered by spatial input, leading to temporal chaos.
So for you listening, this deep dive really highlights the critical nature of timing and feedback loops in the brain, both for building it and for running it smoothly.
And maybe there's a sliver of comfort for people dealing with photosensitive epilepsy, knowing that the trigger can be so specific, tied to the geometry of light itself. It links space and time in a very direct, if problematic, way.
It really does.
Mm.
So wrapping up, these sources lay out incredibly complex temporal programming in such different settings, development,
Right.
pathology.
It makes you wonder, doesn't it? If you zoom out, how universal are these principles? Is the fundamental struggle in neurobiology always about maintaining these delicate feedback loops? Whether it's guiding 40 cell divisions or managing a high-frequency oscillation, is that the core challenge?
A profound question to ponder. That's all we have time for in this deep dive.